up is Aaron Milstein from Rutgers. He's going to be talking about offline memory replay and recurrent spiking neuronal networks. So thank you. Go for it.
Hi, everybody. Thanks for coming so I can share my work with you. I'm going to be asking a particular question today, which is what selects the content of an offline replay event? So I'm also thinking about the hippocampus like Alice was. And so, during the behavior of an animal, if an animal is exploring a spatial environment, then these sort of classical hippocampal place cells will be selectively activated for little snippets of the animal's experience and will be preferring to fire at particular spatial positions. And so these cells will fire in a sequence that's on the time scale of the behavior, multiple seconds. But then when the animal stops to lick a reward or goes back to its home cage to sleep, the hippocampus will emit these population bursts that are called hippocampal sharp wave ripple events. And then some subset of cells will get recruited to spike, and those neurons will fire in an order that often corresponds to the order that they fired during the run phase. But my question is, well, what selects which cells are going to spike in a given sharp wave event, and what decides the order that they fire in? And I'm going to try to address this question with a really simple network model of spiking neurons. So I just have a population of feed-forward inputs. I have a population of excitatory cells that are recurrently connected. And I have a population of inhibitory cells that receives both the feed-forward excitatory input and the recurrent feedback excitatory input. And I won't be able to tell you the details about how I tuned this network model, but suffice it to say that I'm able to get a network model like this to reproduce many of these features of the behavior of the hippocampus. So individual cells can exhibit selectivity for spatial position. As a population, the individual place cells will tile the space, and at different positions, different subsets of cells will be active. And so we can use these sparse representations to try to decode the position of the animal if we have access to all of the activity of these cells. And then the hippocampus during run tends to have oscillatory network synchrony that's in the gamma band that's 30 to 100 hertz and in the theta band that's 4 to 10 hertz. And so I'm also able to get this network to emit theta and gamma oscillations even when the input is arrhythmic. And for the cell models, I'm using a very simple single compartment cell model that's a type of integrate and fire spiking neuron. It has conductance-based synapses. And then the only fanciness is that I can optionally have some spike rate adaptation in these cell models. And so I chose as an inhibitory cell, just sort of a fast spiking cell model so that these cells can kind of maintain high firing rates. But for the excitatory cell, I chose a bursty cell model. So the sort of first couple of spikes that a cell emits, given a constant input, will be at high firing rate, and then the firing rate will tend to slow down. And this helps reproduce this finding that place cells tend to emit these kind of punctuated bursts of spikes that are paced by a theta rhythm. So you'll have to imagine that we're taking a snapshot of this network after some learning process has happened. And so whatever your favorite learning rule is, you can imagine that that's how this network was able to produce its spatial selectivity. And what that looks like is biased synaptic weights over both the feed-forward inputs and also the recurrent excitatory inputs. So a given cell in the network can receive random connections from any other cell. So any of these feed-forward inputs or any of the other excitatory cells. But then what gives a cell a nice sort of signal-to-noise ratio, high firing rate in field and low firing rate out of field, is some biased distribution of synaptic weights. So we've simulated the run phase now. This would be like an animal running down a track. It takes 3 seconds to run the track, and all of the cells are spatially tuned. So then how do we mimic the offline rest period? So this network is inhibitory dominated, and this network is not generating spontaneous activity on its own without any extrinsic drive. So we need to provide at least some kind of a seed of an input to get some spiking out of this network. So I sort of first chose like the most null stimulus that I could think of, which is just to choose random cells from the feed forward inputs to be active together during a sharp wave event. So for 150 milliseconds, there'll be some randomly chosen subset of cells that are just firing at a constant rate. And so because these feed forward inputs themselves were also place cells, each spike is like a vote for a spatial position. And so we can take the spiking activity of this feed forward input population, and we can try to decode a spatial position. Where would the animal had been in space if we had seen this pattern of activity during run? And so you can see, since this was just a random input pattern, the decoded positions are kind of hopping all over the place. They span, they can jump half the track length in a single, in 20 milliseconds. Now, the inhibitory cells in this network, they respond to this random input by self-organizing into an oscillation. So these vertical columns of spikes that you see are the interneurons firing together with high frequency. And then the excitatory cells in this network model, they respond to this input pattern by selecting some sparse subset of cells to spike. And then different cells are spiking at different elements of what looks like a sequence. And so if we try to decode spatial position from this pattern of activity, we see that there are, relatively short hops and sort of spatially contiguous positions are being visited. And this would correspond to like a trajectory that the animal might have been able to run. And so we can do this many, many times. We can have a bunch of instantiations of the network. We can throw 1,000 different, random input patterns at the network like this, and we can look at what the output offline events look like. And so first of all, you can see that all of the positions along the track are visited at some point in some offline replay event. So we get sort of uniform coverage of the track. And then while the feed forward input was just garbage, and so it often had these sort of squirrely trajectories that have big hops and trace out a path that's even longer than the track itself, up to 3 times the length of the track. The excitatory cells in the network are tracing out trajectories that are much shorter and tend to be closer to like half of a track length. And then we can also decode velocity. So, every time bin is 20 milliseconds, and then there's some change in position. So we can compute a velocity of movement. And so many of the offline events have a mean velocity that's positive. So that would be corresponding to a sort of forward trajectory along the track. But then some of the events have a mean velocity that's negative. And so that would correspond to a reverse replay event, or like backwards movement along the track. And what's interesting is that this corresponds to about a 10-fold temporal compression. So in the run phase, it took 3 seconds to go 1 track length, but now we can go up to 3 track lengths in a second. Okay, but then what if we have the network try to learn something salient about the environment? So we know from experiments that if a reward is presented at a fixed location in a circular treadmill like this, that the hippocampus will allocate more neurons to have place fields that are nearby the rewarded site. And so we can mimic this in this network model as well. We can sort of allocate more cells to have their place field locations be nearby a simulated reward. And then we can play this same game. We can play in these random feed forward input patterns. And what we get out is sequences that are, spatially contiguous and tend to visit this overrepresented simulated reward site. And so again, we can do this many, many times, and we see that the positions that are nearby the reward site occur more frequently in these offline events. So this is sort of this network that sort of started off with like a null stimulus that didn't have any structure to it, is kind of already reflecting like the stored memory or the stored experience that's in the recurrent weights and in the feed forward weights of the network. So why is this working? So we can think about some of the previous literature looking at neuronal sequence generation, and something needs to break the symmetry. So one way to do it is with biased connectivity. So you could have a population of cells that corresponds to element A in a sequence, and they're kind of unidirectionally connected to the next set of cells that corresponds to element B. You could do this with synaptic weights too, where you kind of have stronger weights that are going in the forward direction than in the reverse direction. An interesting idea by Sandro Romani was that you could actually use synaptic dynamics for this. So if when a cell starts to spike, it has a high probability of releasing transmitter, but then that depresses over the course of additional spiking, then you can again kind of have some momentum that pushes a sequence forward. And then an interesting study by Kanika Rajan and colleagues had a network that had on average symmetric synaptic weights, but then some interaction between the external input and some fluctuations in the weights were able to drive a sequence forward. So what's going on in this network? So I can tell you a couple of things that will break this sequence generation mechanism. So one is that if I scramble either the feed forward weights on their own or the feedback weights on their own, that disrupts the sequence generation. So there does seem to be some sort of required interaction between the feed forward input and the feedback generated by the network. And then what's kind of interesting is that if I take this bursty excitatory cell model and I replace it with a regular spiking cell model, this also disrupts the sequence generation. So it seems like we were getting some extra benefit by having some spike rate adaptation. And this kind of reminds me of the synaptic depression mechanism, that if you kind of have something different about the beginning of the sequence versus the middle of the sequence, that can kind of add some momentum. But by having symmetric synaptic weights, this network was able to generate sort of both forward and reverse sequences without having like a fixed unidirectional bias. And then the last little bit that breaks this sequence generator is if I just leave out that the objective that this network needs to generate rhythms. So if I tune this network to be sparse and to be selective, but I don't require it to generate theta or gamma oscillations, then the sequence generator also gets disrupted. And so it seems like there is some extra benefit by having the interneurons provide these kinds of intermittent windows of synaptic integration, and that helps to kind of discretize the sequence and add some push forward. So thank you for listening. This work was started when I was at Stanford with Ivan Scholtes, and there were two fantastic Stanford undergrads that helped me with this project, Sarah Tran and Grace Ng, and I have a couple of other great collaborators for other projects. I'm at Rutgers now, and this work was supported by an NIH Brain Initiative grant and an NIMH grant, and used supercomputers that are operated by NSF and by the Department of Energy. Thank you.
Thank you so much for that, Aaron. All right. We have a question, just a sec. All right. So this is a question from Mengsen Zhang. Does the random FF induce a shape wave?
Well, so a sharp wave would be detected like with an extracellular electrode and would reflect, current flowing in dendrites and at synapses and like the spiking activity of like populations of cells. So I don't have that. But I can just take all of the spiking output of the cells and kind of sum them up and look at the total population activity. And that's what I filter to try to look at the oscillations, the theta and the gamma oscillations. And so, yeah, I mean, when you start to provide the feed forward input, then you see this big increase in spiking, and it has a ripple oscillation that's riding on top of it. So maybe that's as close as I could get to mimicking like the actual sharp wave component.
Great to know, thank you. And let me just, all right, close that. And we had another question that referred to Alice's work for you, Aaron. This is from Pierre Orhan. Would you explain Alice's velocity? How would you explain Alice's velocity slowdown in your model? Or
Yeah,
says
actually, I
like
really
he's,
liked Alice's idea that sort of initially you would form sort of a coarse representation of some bits of the environment, but then with additional experience, you'd be kind of refining the resolution and the sort of completeness of your representation. So it's probably that, like, I've taken a snapshot here of this network kind of at equilibrium, like after all of the weights have been learned, but it might be interesting to instead climb in at different parts of the training phase. And look how much sequence generation do you get when there's kind of an incomplete representation after the first lap or two. That's an interesting idea.
yeah, that would be interesting. And I think I may have, there was a clarification to that question. So if you take a look at that question in the Q&A, you can provide any further refinement to that. And we have another question from Diogo Santos Pata. Hi, really nice to see this. Have you looked at phase precession?
Right. So what's interesting is that I build up the template for doing my decoding from the activity during the run phase. but I've also kind of binned it such that I'm mostly trying to capture the spatial selectivity of the cells. But if you do something like, if you really just bin it with really small bins, then you can try to decode position in time intervals that are smaller than a theta rhythm. And then you actually do see that if you try to decode the run phase, that you get these little kind of sweeps ahead and then a reset that kind of looks like something that would be like theta phase precession. And so it could be that, yeah, just by virtue of having sort of rhythmicity that you already get a little bit of this for free in this simple network.